the heart, and we're making this fundamental distinction between the atrioventricular valves and the semilunar valves. But remember, they are all one-way valves.
With valve set in motion passively by an intermittent current of water. When pressure in the ventricle rises, cusps of the valve close together along quite an irregular line.
So you see how it opened there. It opens in one direction only, and these cusps prevent backflow, and that will be true for all of the valves. This is a tricuspid valve. Again, water is being pushed through with pressure.
Each cusp of an outflow valve is shaped like one-third
These are
of
the
a
secondary
parachute.
valves.
Here the cusps are hanging loose. Each cusp has a delicate free border which closes against those of its neighbors. Here's the pulmonary valve in motion.
So it's like it's
In diastole, back pressure closes the valve abruptly, the three cusps pressing against each other to meet exactly at a point. Here's the aortic valve. It works in just the same way. Here's the opening of the right coronary artery, which we'll see in a minute. The left one is out of sight, down here.
And that valve is holding up to dramatically high pressures. We'll talk about the pressures, but again, notice how diaphanous, how delicate it is to handle those pressures. All right, I remember you in high school. You probably dissected a frog or a cat or something, so you're familiar with the term dissection. But I introduced this concept. This is a different meaning of the term, and for some reason, no matter how many times I stress this, we're not talking about the term dissection as you used it on your lab prep cat, frog, or whatever you were using in those days. So please write that down if necessary. We're talking about dissection of the aortic wall, which is a pathology of the aortic wall. Now, if you look at our illustration over here, you can see the heart. We talked about the coronary arteries. We'll come back to those in just a moment. Here are the BCS vessels at the top there. We'll talk about those in more detail later. Sure. But that aorta then is going to arch over and then usually descend on more or less the left side of the midline of the body, vena cava on the right, aorta on the left, into the abdominal cavity. Now, in this particular instance, you'll note that there is an opening up here in what's called the tunica intima. So, like your blood vessels, all the other arteries that you have coursing through your body, the aorta has a laminar organization. It has an interior cellular or layer. There's an intermediate layer, the tunica media, and then it's actually more thicker than this, but they're showing us the outer layer here. Now, what you'll notice is that because of this opening up there at the top, the blood has passed through the tunica intima, that's the white lining that you see there, and has entered this potential space here. Now, I think we've used the term potential space before. What do we mean by a potential space? A potential space is a space that shouldn't be there under normal circumstances. It's a space, we're going to use the term to describe the peritoneum and the pleural membranes and so on. But this potential space, which normally these two layers should be very tightly apposed, they should be right beside each other, but because the blood has gotten in there, it's acted to delaminate the interior and exterior layers. That is what we mean by an aortic dissection. An aortic dissection is a pathological delamination of the layers of the vessel. And I think you can see how it's happened here. You can see the little arrows or whatever those structures are showing us the course of the blood through that opening into that
delamination of the layer
between the two layers. Now, this is a real problem because, of course, there are, as we'll find out later in that descending aorta, there are vessels sprouting off right and left. You have the intercostals, you have the esophageals, the bronchioles, all kinds of vessels. And that's going to present problems. And more importantly, this cannot be sustained. When the blood leaves, what's going to happen as the blood enters this region? Eventually, what's going to happen?
Burst.
Yeah, it's going to accumulate and eventually result in some type of structural failure. Exteriorly, you're going to have massive bleeding, massive hemorrhage. So in the little video down here, this is a surgical repair of an aortic dissection in this region. They can be treated, if you think about it. Where would you rather have your dissection? Again, we don't see it here, but there are vessels popping out all over the place here. That being the case, would you rather have your dissection here, or here, or here? Which would be the most favorable outcome?
None of the above.
No, this is really bad because again, you can't see them, but what about all those vessels leaving there? You don't want to deal with this. This is a nightmare if there's a dissection up there. Your best prospect is right in here. And so that's what is being addressed in this little animation of the video.
This patient was transferred by helicopter with a ruptured dissecting
Clearly,
aneurysm.
Mississippi.
She was taken emergently to the operating room from the emergency room. Transesophageal echo had confirmed the diagnosis. The patient was placed on bypass from the groin. The root of the heart is immensely bleeding.
Now, he said the patient is placed on bypass from the groin. What does that mean? What do you mean bypass?
The heart is
Right, the heart is no
outside.
longer actively pumping large amounts of blood through the body, so they can go to work on it now. That's what they're doing. Of course, the vessel.
You said root of heart, is that right? The area that
Yeah, the root. By the root, he means the region around the base there. The root of the aorta is going to be the base of the heart.
As noted throughout. Hypothermic circulatory arrest is going to be used. But while
So what does he mean by hypothermic circulatory arrest? To lower the blood pressure, to lower the metabolism, lower the efflux of blood. Sometimes in some of these procedures, I think we'll look at one later, they just throw ice in there, just dump the ice chest in there.
the patient is cooled, the aorta was clamped so that perfusion would be adequate. Subsequently, the clamp was removed distally. The intima is removed inside of the aorta. The two layers are reconstructed using felt. A HemoShield Dacron graft is sewn into position distally. It is clamped and then perfusion is begun again.
So a stent is used.
The coronary arteries are going to be connected with a Dacron graft. The Gibraltar.
See, the problem you do have up here, you can't really see them too well in this figure, you have your coronary arteries up here too.
Technique. A St. Jude valve conduit has been sewn into position at the level of the aortic root. The two grafts are connected. At the conclusion of the procedure, cardiac action is excellent. The coronary arteries were anastomosed to the main graft coming out of the heart, and both were perfused through this Dacron graft. The valve functioned well. It took some time to get good hemostasis, but the patient did extremely well and is seen here at home 3 months later, attending to her plans.
Isn't that remarkable?
That's what we needed.
I think you will find in your experience that aortic dissections generally do not have a good prognosis, and it just occurs, it depends on where they occur. But as far as I know, that's about the only place that they can occur that they're not fatal. Yes, you.
Yeah. Would you repeat why it's better to happen near the base of the heart?
Yeah, because again, you don't see it here, but remember when we talk about this thing is coming up over here, right around this level, and then the aorta is going to descend toward the diaphragm. And as it does so, it's giving off those thoracic arteries in both directions. It's giving off thoracic arteries, which we call intercostal arteries. It's giving off bronchial arteries, esophageal arteries, just a whole spaghetti. All right. So think about what's going to happen with delamination to all those vessels. You're going to have bleeding around them, from them. How would you correct that? You can't put a stent in there. You'd have to put stent that put holes in it. That wouldn't work. It'd leave you where you. So yeah, if you're down in this region, I don't know of any mechanical solution to the vessels. I can tell you what they typically do just based upon my reading, but it's only a short-term solution.
Are there any symptoms?
Yeah, for example, this is more common in obese patients, it's more common in smokers, it's more common in individuals who have vascular pathology of some other type. Sudden loss, I mean, they have, if it's a severe delamination or dissection, problems breathing, chest pain or referred pain, weakness, lethargy, it's fairly, they're not usually capable of continuing normal activity. And of course, if it's not caught very quickly, fatal. I've known a couple of individuals who passed from this particular problem. Both were smokers. All right, we've talked about the histology of the cardiac muscle, so let's just point out a few other features. Remember, we said that in skeletal muscle, the fiber is the cell, and skeletal muscle is very long cells, basically extend the length of the muscle. They're multinucleated in skeletal muscle. We talked about the fact that they have a sarcomeric organization and so on. So in some respects, cardiac muscle is similar to skeletal muscle. In other ways, it's rather different. Cardiac muscle is striated, means it has the sarcomeric organization that we saw in skeletal muscle. It has Z lines and M lines and all of that. However, the cells are mononucleated, at least usually that's the case, instead of those DNA units that we talked about in skeletal muscle. The cells tend to be shorter, narrower, and you'll notice that as they're organized into what is sometimes called a lattice work arrangement. By lattice work arrangement, you can see that they're parallel at some points and then they separate and then they'll come back together. And so it's sort of a lattice work with respect to how they extend beside each other. There's a much lower abundance of transverse tubules than in skeletal muscle. And we'll talk about some of the consequences of that as we talk about the action potential. And of course, functionally, physiologically, the energy demands of the energy demands of cardiac muscle are much greater than skeletal muscle because this can't take a break. This can't, like a smooth muscle, it has to be capable of repeated contraction at a very intense and ongoing rate. Consequently, both adequate levels of energy and oxygen must be provided to the cardiac muscle. And we'll talk about the specialized blood vessels that are responsible for providing that oxygen and nutrients, the coronary vessels. And then we'll talk more about the nature of the contraction in just a moment. We indicated earlier that the individual cells are joined by these intercalated discs that you see here, the sort of squiggly lines. And those intercalated discs are redolent with both desmosomes. Remember, those are the cell junctions that weld adjacent cells together, and gap junctions that that enable depolarization in one cell to be rapidly communicated to an adjacent cell. So if we're taking a portion of the myocardium here, you can see it blown up in this illustration. You can see the lattice work, mononucleate organization of the cardiac cells. And then as you look at just a portion of the intercalated disc there, that's what we're looking at here at the bottom. This structure, it sort of weaves and bends like a meandering river, but it includes both desmosomes and gap junctions. Otherwise, you see the sarcomeric striations. And if you look closely, even in this graphic, I think you can see some of these little blue lines here. There's some there, some there. Those are the intercalated discs. Kind of sort of see them there. All right, so now we need to crank it up a notch and talk about the cardiac action potential. By the way, what do you notice? Can you see those cells there? This is an animation. This is a video, and this is a Petri dish full of cardiac myocytes, contractile myocytes. And what are they doing that you notice there? Yeah, they're dancing. These cells can contract on their own. They do so in a rhythmic pulsatile fashion, and they require no outside stimulus to do that. So that's going to be a very, very important characteristic that we need to talk about. Now, we're going to simplify this discussion dramatically. We can classify the cells of the heart in a variety of ways. for the most part, in fact, you can see down here, we could talk about the atrial myocardium, the ventricular myocardium, we could talk about pacemaker cells and conductive cells. We could organize our discussion in that fashion. But really what I'm going to do, to make this very simple, we're going to divide or suggest that there are really only two types of cardiac cells, right, to make it very simple. There are the myocardial contractile cells, which are responsible for the contraction in the wall of the heart, and of course it's that contraction that's responsible for the pumping of the blood. So those are the contractile cells that are present in the myocardium. In addition, there are pacemaker cells, and let's just call all of these cells that are responsible for electrical conduction, we just call them conduction cells, and they're going to deliver an electrical signal to various parts, various regions of the heart in a very specific fashion, in a very time-dependent fashion. Now, part of those conduction cells are the pacemaker cells. And so we're going to look first at these pacemaker cells, understanding that their function in the heart is to serve as the clock, to serve as the metronome, to serve as the timekeeper, responsible for indicating when the contractile myocytes should contract. So let's make sure we understand the general logic of the ensuing discussion. Specialized pacemaker cells in the heart, and we're going to talk about where they're located, especially in, for example, the sinoatrial node, serve as timekeepers, sending out electrical signals, action potentials, that are then propagated along the conduction pathways, for example, from the SA node to the AV node and down to the ventricle. But that signal is going to be myogenic, or another term we might say intrinsic. So in other words, the heart, the pacemaker regions of the heart. And this gets a little complicated, but we'll begin with the SA node. In fact, all of our discussion will pretend as if the pacemaker cells are exclusively in the SA node. Will be responsible for generating an electrical signal intrinsically, independently of any other type of stimulus that is then communicated to other regions. So let's begin, as I say, let's begin our discussion with the, again, I've broken my computer. Let's begin with the pacemaker cells. So just be sure you understand that this slide refers exclusively to what we're calling pacemaker cells, and those pacemaker cells that we're going to be talking about are in the sinoatrial node, the SA node. And then we'll expand upon that. And once again, the function of these cells, my poor computer just is not functioning today. These pacemaker cells are responsible for generating that metronome or rhythm to which the rest of the heart should respond. All right. Now we've talked about neuronal action potentials, and remember we've talked about the resting membrane potential. So all these terms should be familiar to you. When we talked about smooth muscle, we also talked about a pacemaker potential. Remember when we talked about smooth muscle pacemaker potentials, we said that if the pacemaker potential, which is an oscillating, vacillating membrane potential going up and down, up and down, up and down, if it reaches that dash line there, which is threshold, then the pacemaker potential will initiate an action potential. Okay. So in pacemaker cells of the heart, which are located where, at least in the SA node. All right, so in pacemaker cells of the heart that are located in the SA node, the cells generate or are capable of generating a pacemaker potential, as we saw in smooth muscle. That it reaches threshold, results in generation of a pacemaker action potential, which is then communicated down those conduction pathways. So we have two types of potential that we need to keep straight in the pacemaker cell: the pacemaker potential and the action potential. So again, this portion for right now, this portion is the pacemaker potential. It just goes up and down, up and down. This is the threshold potential. And so what is this?
Action.
That's the AP, all right, that's the action potential. So we don't really see the full pacemaker, but you can draw it in yourself. It would be something like that, especially if I were capable of drawing something like that. You can do a lot better job than I can. Now, as you look at this illustration, if this is the pacemaker potential here, and we'll talk about how this happens in just a moment, but let's do this one step at a time. What is the lowest or the most negative potential of the pacemaker potential? About -60. And it peaks at about -40. So it oscillates between -60 and -40. By convention, and I'm not too concerned about this, but by convention, we say that pacemaker cells in the SA node do not have a RMP. They do not have a resting membrane potential. Why? Because they leak. They leak. And we talked about potassium leak channels. These cells, the pacemaker cells generating the pacemaker potential, leak sodium. Isn't that a kick? They leak sodium. So if they leak sodium, if we start out at -60 here and they start leaking sodium very slowly, just slowly leaking sodium, what's going to happen to membrane potential over time?
Increase.
Slow depolarization up to about -40. Now, as this is happening, as we're going to call them for a moment, let's just call them sodium leak channels, leak, another type of channel is going to open. So as we move from -60 to -40, another type of channel, a voltage-gated channel, is going to open. Remember what causes voltage-gated channels to open? Voltage-gated channels open in response to a change in membrane potential. Voltage-gated calcium channels open. And if the pacemaker potential reaches threshold, the relative abundance of calcium channels that opens increases dramatically, such as you see this action potential. So what we're saying here is the pacemaker potential, well, let's make it very simple. For both the pacemaker potential and the action potential of the pacemaker cell, it is primarily calcium current that is responsible for the depolarization. I'm going to qualify that in just a moment, but let's just make that a rule first. For both the pacemaker potential and the action potential of the pacemaker cell, it is chiefly the calcium current that is responsible for the depolarization. Chiefly. But what did we say kicked it off down here? What type of channel? The sodium channel. All right, so we're going to come back to that sodium channel in just a moment. And then what tends to happen, this should always occur. So if the pacemaker cell is functioning properly, we should always have the pacemaker going back and forth like this. And as it does so, it's generating these action potentials at the rhythm of oscillation. And this action potential then will be propagated along the pathway we'll talk about later, from the SA node to the AV node and down the Purkinje fibers and so on. All right, these voltage-gated sodium channels are going to close on their own. Remember, in general, all of these voltage-gated channels only open within a narrow window of voltage. They open in response to a change, but they only open ideally in a narrow window. So as we move away from that -60, they're not going to want to stay open so long, and that's in part responsible for what causes the repolarization of the pacemaker.
Is it the calcium channel that is open?
Yeah, so for now, I'm just want to try to do this because
Yeah, it
it's a,
confuses.
it's a feedback process. And anytime we have this type of feedback, we have to find a point to begin. So my beginning point is to have you understand that for most of this depolarization in the pacemaker and the action potential is chiefly calcium. All right. But our problem is, and I think I'm anticipating your question. So here we have cessation of the the sodium leak channels. We have repolarization, which in part is mediated by potassium channels, which we learned about in the neuron. And so this is going back and forth. But when the membrane potential begins to repolarize and approach -60, that's when that sodium channel opens in earnest. So this is the tricky part, and this is why we call this channel, and this is what we all have trouble with. This is called a funny channel. The funny channel is the HCN channel, and I think I have it written there at the bottom. Excuse me, my computer's just right down here in the very fine print. If you look, the leak channels, the sodium leak channels, are the HCN channels, and the F here stands for funny. So let's figure out why they're funny. They're funny for a couple of reasons. They're funny because, you tell me, the sodium channels, are they depolarizing or repolar? Are they depolarizing or hyperpolarizing? They're depolarizing. So when these sodium channels down here is where we're talking about, when they open, they depolarize the cell membrane.
Mhm.
But here's my question for you. That was question number one. What causes them to open? What causes those funny channels to open, these depolarizing sodium channels, which are also leak channels? What caused them to open?
Change the membrane potential.
Change in membrane potential. More specifically, more specifically, let's keep it very simple here. More specifically, it was the hyperpolarization that caused the depolarization. So funny channels open in response to a more negative movement of the membrane potential toward -60. So the closer we get to -60, the more likely those funny channels are to open. And when they open, unlike the voltage-gated channels we've encountered previously, they remain open for a while. They remain in this sort of leak. That's why we get to call them slow sodium channels. So they're funny for two reasons. Why are they funny? Why do we call them funny? Unlike any other channel we talked about, they number one, they depolarize in response to hyperpolarization. They open close to -60 here if the membrane potential is coming from above. Shouldn't use words like above, but from a more positive direction. What's the other reason they're funny? What makes them funny? They leak. So these are voltage-gated channels that don't follow the general premise that voltage-gated channels open for only a few 10,000ths of a second and then snap shut. They tend to leak. So it is this concerted interaction of sodium channels, the funny channels, the calcium channels, and the potassium channels that are responsible for this pacemaker potential that occurs only in pacemaker cells, which for the current discussion are present exclusively in the SA node. Now, with respect to the AP, this is a little bit different too, because the depolarization is entirely calcium. There's no sodium here. So the pacemaker cells, the pacemaker cell action potential, depolarization is entirely mediated by calcium. And then we have our good old buddy, the potassium channel, that's responsible for repolarization. So very different sort of animal. And nothing's working. But in any case, what this animation would have shown would be again, the rhythmicity. It's called autorhythmicity of the pacemaker cells.
That's very confusing.
All right. So now, make sure you notice we're changing our discussion entirely now. Now we're no longer talking about pacemaker cells. Make sure you appreciate in this slide, we're talking about contractile cells, the contractile myocytes that are present in the walls, the myocardium of the heart. Now, if you look at these, they started around, they do have a resting membrane potential, and it's about -90, -95, so it's much more negative than about any other type of cell we've talked about. In general, muscle cells, both skeletal muscle and cardiac muscle, have a more negative resting membrane potential than would neurons. So here's our resting membrane potential indicated by the black there. And traditionally, this is called phase 4 of the contractile myocyte action potential. So phase 4 is the resting phase, the RMP there. Then, again, very different than the pacemaker potential, there's going to be rapid depolarization, signal-dependent depolarization caused by sodium channels. So this depolarization here that you see here in phase 0 in the contractile cell is entirely mediated by sodium. Notice it only lasts a few milliseconds. Then, as we have discussed before, the voltage-gated sodium channels, because we're moving away from their optimal window of opportunity, they're going to start closing very, very rapidly. This is all occurs in just a few milliseconds. Then those voltage-gated potassium channels, and we're going to talk about the different types of channels, but up here there's a voltage-gated potassium channel that opens, that initiates or completes the repolarization, or doesn't complete it, initiates the repolarization. And then something odd happens. And you've encountered this before, except there you called it a DHP receptor. Do anybody remember the DHP receptor? It was a slow type L calcium channel. So at about this point right in here, calcium channels open, the slow type L calcium channels. And as they open, those potassium channels are still open. So potassium channels, are they depolarizing or hyperpolarizing? Everybody remember potassium? Hyperpolarizing. Calcium, is it depolarizing or hyperpolarizing? So we've got a battle. We have a battle between the calcium and the potassium. Remember, potassium channels are still open here. We're going to talk a little bit more about these channels in just a moment. But our initial simplistic presentation argues that now the depolarizing calcium is working in opposition to the repolarizing potassium. What is the result? A standoff. And the membrane potential remains fairly constant until it reaches about 0 with a steady decline. That's called the plateau phase. The plateau phase is phase 2 of the cardiac myocyte action potential. And it lasts typically about 175 milliseconds. And when the membrane potential slowly declines to about 0 millivolts there, about that point, the calcium channels close and the potassium channels, or a specific type of potassium channel, remains open and repolarization resumes. And then that takes us back to the RMP. And that phase is phase 3. You can see this thing is not quite drawn to scale because the repolarization phase is actually considerably shorter than the plateau phase. And then we start back at phase 4 again with the resting phase, back around 90 to 95 millivolts typically. Now, this is a delightful little figure here. But I think you probably understand in general what's going on. Here's our resting membrane potential here and here. Here's the depolarization, repolarization, and so on. In addition, you'll notice that there is just a delightful array of channels. Each one of those little boxes is a special type of channel. I stands for current. I think it's German for Intensität or something, but that's the traditional uh, abbreviation for current. So what is indicated by this symbol right here? What type of channel is this? That would be a potassium channel, because a potassium channel would allow potassium ions to pass through, and that would constitute potassium current. All right, what would this one be? How would you describe this? What can you make of that symbology? Excellent. So we've got several things to work out here. We've got a potassium current, and you know ACH2 stand for acetylcholine, the heart. The force of contraction, remember our discussion of the autonomic nervous system? Is there any relationship between the autonomic nervous system and your heart? What this is a potassium channel. It's basically a muscarinic receptor, or at least it responds to, it's coupled to a muscarinic receptor. The channel is coupled to a muscarinic receptor, and the point being that this is one mechanism by which the autonomic nervous system regulates the force and timing of heart contraction. The autonomic nervous system has a direct effect, among other things, upon how long, in this case, the potassium channel remains open. Of course, we're talking, is it clear to you that these two boxes refer to this? And then, what is this? Yeah, the little arrow means that the current is coming into the cell. It's a sodium current coming into the cell. Here we have TO stands for transiently outward. Transiently outward. That's a potassium channel again. Why is it outward? Which way are the potassium ions going? When the channel opens, which way are they going, in or out? Out. But only briefly. And you know it's briefly because it doesn't last very long. Then we have, by the way, I'm not asking you to remember all these channels. I just want you to think about the complexity of this. This is another potassium channel. UR stands for ultra rapid. Because we have an ultra rapid response, an ultra rapid repolarization or hyperpolarization. And then you already know what this one is. What is this one? The depolarizing L slow type calcium channel, which is, again, battling with the potassium current. KR is rapid. I don't remember. Anybody remember KS? I'm having a mental block. It's not short. Slow. Thank you. Thanks. That's exactly what it is. So you can see that we have a couple different families of potassium channels helping to regulate. And by the way, what was learned in recent years is both these K and the KS and KR are also responsive to autonomic innervation. So the rate of repolarization is also going to be influenced by autonomic signals. So you get the idea. I just want you to see another IKACH, another point at which the autonomic nervous system can regulate the timing and force of contraction. Here, this thing down here is a sodium-calcium exchanger. We learned another term for that. If sodium is involved in exchange, what type of transporter is it most likely to be? A secondary active transporter. That's all this is. Exchanging calcium for sodium. Sodium's coming in, calcium's going out. I think there's a 3-to-1 ratio between the two. But the point I'm making, I'm not going to ask you to learn this. This you should know thoroughly. These phases and this you should know thoroughly. But I just want you to understand as you look at this illustration that as a PA, there are many points of regulation, both autonomically and intrinsically and within the heart, to regulate the timing and force of contraction in the contractile myocytes. Keep in mind, we're only talking about those contractile myocytes that are present in the wall of the heart here, the ventricular wall and atrial wall. We'll come back to this later when we have a little more knowledge. At the top here, we're looking at an EKG trace, EC or EKG trace. And you can see it's lasting about a second there. And then what is this? Somebody tell me what that is.
Action potential.
What type of cell? All right, this is the contractile myocyte. You would not confuse this with a pacemaker cell. There's no way that could ever happen on a quiz or test, right? So we're looking at the contractile myocyte. And the EKG, we're going to learn a little later. This is called the QRS complex. You can see it, at least in the lead 2 of the EKG, there's this peak, a pointy peak there. To what does that correspond in the contractile myocyte action potential? Depolarization. So really, yeah, ventricular depolarization. There's going to be one for the atrium too. So as I say, we'll come back to that. You'll also notice the difference in magnitude over here. We're looking about 1/10 of a volt in the action potential and only a millivolt in the EKG. Why would that be? Why would the EKG be so weak by comparison to measurement of the action potential, do you think? Yeah, in the top illustration, we're measuring voltages, signals that are a long way from where they originate in the heart. Now here, this is an imaginary voltage, as if we actually had the little electrodes right across the cell membrane. So much, much larger. Another illustration, of course, whenever I do this, this means that you need to be very familiar with it. You need to know the general sequence of events. By the way, I have a little figure down here and also have something labeled refractory period. What was the definition of refractory period? We talked about it in skeletal muscle and in the neuron. What is refractory period? Right, it can undergo another depolarization. So notice that the refractory period for the cardiac contractile myocyte is approximately the same as the cycle of depolarization and repolarization. It's a little shorter, but what that means is, and this is kind of a safety feature built into your heart, is that once that myocyte is undergoing depolarization, it can't undergo another simultaneous depolarization, and that's kind of fortunate because that might lead to something like flood or fibrillation or something like that. This is one of the many safety mechanisms we'll point out. And you have the same illustration here. So in other words, here the red is the myocyte action potential. The entire period of the yellow here is the refractory period. So that cell is not going to be able to generate another AP until it's completed the first AP. And the blue is the contraction. So what do you notice about all three of these? The AP, the mechanical contraction of the cell, and the refractory period are pretty much concurrent. They're pretty much simultaneous, not precisely, but pretty much overlapping there.
I appreciate this little pictures here.
And that's in contrast, for example, skeletal muscle where you had the AP and then you had the delayed contraction, and that would go on for about 1/10 of a second. Notice in cardiac muscle, again, all three, again, for our simplified purposes, pretty much overlapping, and the interval is much longer. 250 milliseconds is what fraction of a second? Yeah, a quarter. So we're going to talk about the timing in more detail next time, but this is about a quarter of a second. All of this can be completed.
So
That's a good point to quit.